So please feel free to interrupt me at any time. Today we're going to talk about quantitative MRI with a special focus to the spinal cord, but at the end of the day, you could apply it to any parts of the body. We're going to start by describing some of the features of the techniques that we can do with advanced MRI techniques. Then we're going to dive into the specifics of spinal cord imaging, and we're going to see why it's a challenging area for obtaining proper images. And then we're going to check whether we're going to find ways to validate those quantitative MRI metrics using histology. So first of all, the context that I always show is that's a patient that was referred to a neurologist at MGH in Boston, who I was working with. It's a patient who unfortunately had a lesion, intramedullary lesion that was produced by a clinical malpractice. Basically, they injected some corticosteroids inside the medulla where it was supposed to be around it, and as a result, this patient had a very focal spinal cord injury that we can see on the T2 weighted image. But this patient had some chronic motor and sensory deficits that could not be explained by the location of the lesion. So we assumed that there was some degeneration that happened in her spinal cord white matter. And unfortunately, we could not see those abnormalities on the conventional MRI. So that kind of sets the context for the so-called quantitative MRI. What's interesting with those techniques is that we are able to obtain additional information about the underlying microstructure of the central nervous system and potentially, other structures. So people apply that to the heart as well, for example. And there's, as you can see, a lot of techniques. You've probably heard about diffusion MRI, but there is also other techniques that enable to get this information. In addition to microstructure, which is more of a static assessment of the structure, we could also gather insights about the actual neuronal activity of the neurons. And this is done indirectly through changes in blood oxygenation with a technique called fMRI. We could also have access to metabolic information. And ultimately, all those fancy techniques are used for improving diagnosis, prognosis of patients, and also provide pharmaceutical companies with an objective marker for testing new drugs, for example. So now you probably all know basics of MRI, how it works, and we were talking about microstructure. So let's take a nice brain. It's actually a full brain that was sliced by Alan Evans' team and produced this wonderful large-scale histology that has a resolution of about 20 micron. And let's do a little game of downsampling this to the resolution of an MRI, and it's actually a pretty good resolution already. It's like 500 micron, and we see that we have those ugly big squares that are supposed to represent this rich microstructure in there. So, is it really possible to see microstructure with MRI? So we're going to dive into some of the techniques, not all, of course, but some of the, I would say, popular techniques that are used, so that you'll have some ideas about how it works, how we can get information from each of those voxels. The first one is called DTI. How many of you have heard about DTI or know how it works? Okay, so it's not a bad idea to go through that. It's based on the properties of water diffusion. Water diffuses, like the water molecules are always in constant motion due to Brownian motion, and if we drop some ink on a Kleenex, you'll see that at some point it will become, like a circle. and we call it an isotropic diffusion. Of course, my screen is kind of squeezed, so it looks more like an isotropic, but let's assume this is a circle. However, if you drop this ink on a newspaper, which has fibrous structure, the water will tend to diffuse preferentially along the fibers of the paper, right? We call it an anisotropic diffusion. What's interesting is that if we place, if we do this experiment in a celery, for example, which has a lot of fibrous structure along this axis, we'll see that the water tends to diffuse along this direction. The good news is that our brain is a bit like celery, and especially the white matter, because the white matter axons are packed bundles. So the axons are the elongated parts of the neurons that enable to transmit action potentials. If we zoom in, we see that those axons are composed of the intracellular part and also this kind of purple-ish part, which is called myelin. And myelin is kind of a barrier to the water molecules, so they tend to diffuse preferentially along those axons, right? So, okay, that's the property of water diffusion in the white matter. Now, can we measure that with MRI? And the answer is yes. So I'm not going to dive into the pulse sequence aspect of it, but with MRI, you know that we have the system has gradients, and those gradients are used to encode the spatial location of the spin, so that we can reconstruct an image. We can also use those gradients to encode something else. In that case, we want to encode the direction of water diffusion, right? So what you're going to see here is that those gradients will be applied along these arrows. And I'm going to turn those arrows in circle, right? And what you're going to see is that the recorded MRI that has those encoding gradients will have will look different depending on how these gradients are oriented. So I'm going to turn it now. And you could see that, the signal, especially in the corpus callosum, which is, highly packed bundles of axons, changes its signal, whereas in the other parts, like the gray matter or the cerebrospinal fluid, the signal is not really dependent on the orientation of those gradients. So in fact, we're able to sensitize the water diffusion thanks to those magnetic gradients. And we can put that into equation and extract the part that really explains how the signal varies with the application of those diffusion gradients. So how does a typical diffusion tensor imaging protocol work? We acquire several of those images with different gradient orientations. We acquire one without any diffusion sensitization with the gradients. Then we process them all together, and we fit some models. There are different models that exist. The most common one is called diffusion tensor. And it's a 3x3 matrix, so it's a fairly robust fit that we do with all those images in each voxel of the image. And then if we map this diffusion tensor, you basically have a map of how the water diffuses along in each of the voxel of the brain. And then from this tensor, we can also extract metrics. You might have heard about fractional anisotropy, or FA, which explains, how spherical or how ellipsoid this tensor is. And, the more ellipsoid it is, and the more the water will diffuse in a particular direction. Okay. So, for example, in the white matter, those tensors are very squeezed, almost like a line. Whereas in the cerebrospinal fluid, where the water has no barrier, it can diffuse in all directions in space. The tensors looks more like a big sphere. So some computer vision people, played around with that, and then they came up with a method called tractography, where the idea is to basically connect, starting with some seed points, and then connect all the neighboring tensors that have similar orientation, right? So we can see it everywhere in the brain, and then obtain those kind of, nice-looking tractograms which represent the white matter architecture in the central nervous system. Of course, this is a tensor diffusion tensor is a simplistic view of how water molecules diffuse, because the central nervous system is a complex structure. There is a lot of crossing fibers and fanning. There is also different properties of the microstructure. So DTI only cannot explain all of it. That's why there is ongoing research that tries to model the different compartments of the white matter, and especially the inside compartments, which is called the restricted compartments, where the water is kind of trapped inside the intracellular space, and the outside compartment called the hindered or the free compartment. And so that's the theory, and then we can devise some MRI experiments in order to measure, basically have like a volume fraction of those two different compartments. The way we do that is similar to standard DTI protocol, where you have some gradients, like on each side of the 180 degrees refocusing pulse. So that's a typical spin echo with diffusion encoding gradients. But what we're going to do is that we're going to vary those amplitudes and also what we call the mixing time on each side of the 180 degree pulse. And if we do that, we are able to fit some models, and there are a lot of models that exist. There is the CHARP model, the active axe, NODI, TEDI now. So there's a lot of models that exist, more or less complex with more or less parameters. But ultimately, the goal is to really get the volume fraction of the restricted diffusion, hindered diffusion. And why is that? What can we do with this information? One thing we could do is then represent those different volume fraction and estimate how much of axons we have in a particular volume. And what's cool is that we can also infer on the actual shape of those axons and size. For example, we could infer on the mean axon diameter. So that was demonstrated in a rat corpus callosum, and they were able to show that what they inferred with MRI in terms of axon diameter distribution matches what they observed from histology. So, this looks exciting, and there's a lot of ongoing research in this direction. If you go to the main conference in MRI research called ISMRM, there will be a lot of sessions and talks about that. So we cannot forget one element, which is myelin, because when we do diffusion imaging, the echo time is usually very long, and we get no signal from the myelin. But it's an important component because it can be affected in neurodegenerative diseases such as multiple sclerosis. So myelin is composed mostly of lipids, and it also has proteins. And its main function is to accelerate action potential communication. And as I mentioned, it is affected in different diseases such as MS or NMO. So we would like to be able to, in addition to measuring the density of axons, their shape, we would like also to measure the concentration of myelin. Can we do that with MRI? Well, there are different techniques that exist. Again, I'm not going to go through all the techniques, but let's talk about magnetization transfer. The main idea of magnetization transfer effect is that with MRI, that most of the time you excite the water protons with a particular frequency called the Larmor frequency. At 3 Tesla, it's about 127 megahertz. And it's a very sharp excitation profile, right? But the thing is, myelin are made of macromolecules, and those macromolecules have a much wider resonance frequency. So the idea of magnetization transfer is that we'll excite at an off resonance pulse, so not at 127 megahertz, but slightly below or above. and that will basically excite those macromolecules, which will then transfer their energy to the water molecules that are close by. And they will be kind of pre-saturated. And when we do a regular imaging, the signal in those water protons will be attenuated. So one idea was to do two experiments. One experiment with normal imaging parameters, and the other one is called with this MT preparation pulse, where we will excite at an off resonance. The water molecules that are close to the myelin will basically be pre-saturated, and then when we record the image, when we record the signal, the signal in the white matter will be slightly lower, right? So the idea is to take the ratio between the two and obtain a score called the magnetization transfer ratio that was shown to correlate with myelin. Now it's correlation, but it's not like a 0.99 correlation. There is other effects that could drive this MTR, notably the T1 relaxation, which might be different depending on the location. Also, the B1 plus the excitation profile is not perfect, and it could also affect the MTR pulse. There are better ways than MTR to basically try to account for those effects, called MTSAT. And if you want to go, a little further, you could also try to model the different compartments that are responsible for the magnetization transfer exchange. And Yves is actually a world expert on that, so could tell you a lot about this. So that's one technique. There is another one, which is fundamentally very different. The idea behind this techniques, which is called myelin water fraction, is to estimate the proportion of water that is trapped inside the myelin sheath. Right? What's interesting with this water is that it has transverse relaxation time, so a T2 decay, which is faster than the water that is inside or outside the axons. That's because of the interaction with the myelin. So why not trying to divide an experiment where we acquire a lot of echoes, and you've probably been told that the standard relaxation T2 relaxation is a mono-exponential decay. So you acquire a couple of points, you fit a mono-exponential. In that case, it's not mono-exponential. It's if we force the model to be a bi-exponential decay, then you will try to fit two exponentials within this curve. So now you have a bit of a technical background, so you know that it could be problematic if the data are noisy, because it makes the fit less robust. What's nice with this technique is that it's specific to myelin compared to measuring an effect that comes from the macromolecular from the macromolecules. But as I mentioned, it could be noisy, which which could yield some inaccuracies in the measurements of the myelin water fraction. It's also long. You need to acquire all those echoes. But there is also ongoing research, very recent research on other techniques that try to acquire faster, that rely on other relaxation than the T2. Again, with the ultimate goal of measuring the myelin water fraction. So, if we want to summarize the myelin mapping techniques, there is, we talked about MTR. There's QMT. There is another one called inhomogeneous MT. We talked about myelin water fraction. Some people also do T2 star mapping, T1 mapping, and all of these, have more or less specificity to myelin and more or less confounded by other factors. There is also, people try to combine different methods, and so each year at ISMRM there is, new techniques that comes out. So now you have a bit of information about what we can measure in terms of microstructure in the central nervous system. Is that easy to apply those techniques to the spinal cord? So that's what we're going to look at now. If any of you have any question at this point, please feel free to ask. Okay. So the spinal cord is small. So, we don't have infinite amount of SNR, of signal-to-noise ratio. So it's always a compromise. If you want to image a small structure, you need to reduce your voxel size, but this comes at the cost of SNR. What can you do to increase the SNR?
Increase time.
Yeah, increase time, but you're in a hospital here, right? So you know that time is not infinite either.
Coils.
Coils, yeah, exactly. And it's, I wanted to say it's a cheap way of getting SNR, but there's a lot of trends for going to 70 and things like that. But before going to 70, get a good coil, and it's cheaper, and you could get.
By an order of magnitude.
Yeah, yeah. So better coils are help a lot to gain sensitivity. So there is a lot of research for what's the best coil that we can make for a particular area. Of course, if you have money, you can also get like a stronger magnet, but that also comes with the cost of more challenges in terms of V1 plus homogeneity, in terms of SAR, in terms of susceptibility artifacts. So if you can combine those two, you really get the best pictures. So this brings me to challenge number 2. So this is, I showed you before the concept of tractography, where you try to reconstruct the white matter architecture using diffusion imaging. So this is a tractogram that is overlaid on an anatomical image. And it's interesting because we see that the tractography stops at some point. So we could conclude that maybe this person has a complete spinal cord injury. In fact, I was the subject. So I don't have any spinal cord injury, and the reason the tractogram stops is simply because there was some susceptibility artifacts on the image. Of course, we don't see that because I'm not showing you the EPI that was used to acquire those diffusion images. So be very careful when you read papers on diffusion and tractography, because sometimes interpretations are a bit quick. So if we correct, we can correct and we can do some post-processing of those distorted EPIs and correct for the tractogram and have something that makes a bit more sense. But this really brings this issue of susceptibility artifacts, and it's due to some errors that are accumulated when, especially when we do EPI, echoplanar imaging, which is a fast way of getting images, and it's very popular for diffusion and for fMRI. And it's particularly problematic in the spinal cord region because there are different structures with different susceptibility profile, and that creates a local gradient, a local magnetic gradient, and that is bad for API because when we do imaging, we assume that it's on the 3 Tesla scanner we have 3 Tesla everywhere, but because of those different profiles, we have 2 point something at a particular location and 3 points epsilon at another location. So we don't have 3 Tesla everywhere, and that results in those distortions. There's another effect which we call dynamic B0 offset. And when you're in the scanner, so usually we image living people, and those people breathe, and when they breathe, they fill their oxygen, they fill their lungs with oxygen, which is slightly paramagnetic, and since this is paramagnetic, it will also have an impact on the B0 field. And this, basically you end up having a varying B0 field with time. So what you see here is that we made an experiment when we asked a subject to go in the scanner to inspire, to keep their inspiration for 10 seconds, and we acquired some EPIs. Then we asked the subject to expire. We acquired another EPI, and what you see is basically the back and forth between those two EPIs. And I can guarantee you that I'm going to demonstrate that after. I can guarantee you that the motion you see is not the motion of the patient when the person breathes. It's simply due to a shift in the B0 field, right? And the phase encoding was in this direction, and you can see that the motion appears in the phase encoding directions. So that's absolutely horrible. There are ways to tackle that. One way is to work on the full sequence, basically trying to acquire the k-space faster so that we minimize the accumulation of error. And you might have heard about parallel imaging, which is a way to acquire the k-space faster. There is also reduced FOV technique, which is similar to the parallel imaging approach, but with reduced FOV, we basically will try to, instead of acquiring a square, we'll reduce the FOV in this direction, and therefore we can reduce the number of phase lines in the same directions. By reducing the amount of phase lines, we feel the k-space much faster. Another approach is to address the problem at its source, basically trying to make the field more homogeneous before doing the acquisition, or like during the acquisition. And that's a procedure called shimming, which is always done when you acquire an image. Here the scanner goes like bzzz, and it's trying to find good combination of currents in the shim coils that are in the system to make the region of interest, like for example this region, like most homogeneous possible. But that addresses the static homogeneity aspects. What do we do with this dynamic susceptibility effect? So I'm going to present you the work by Ryan Topfer, who is in the room somewhere. Yeah, right here. And so Ryan is working on an external shim coil that was built by Resonance Research in Boston. And the idea of this shim coil, so it's a 24-channel coil, on top of which we designed an RF coil, transmit-receive, because we could not use the body transmit for that, with that amount of copper, of heavy copper. And on top of that lies the subject. So now the setup that we are working on is we have the subject on the patient table with the 24-channel shim-only coil and the 8-channel transmit-receive. Those shim coils are connected to an amplifier that sits in the equipment room, and those amplifiers are driven by boards that are controlled via a serial system, and basically this connection is related to the control room. So in the control room, Ryan sits there, and he receives information in real time from the patient's respiration. So we built an absolute pressure respiratory bellow that is connected to an Arduino. This Arduino connects to the laptop, so in real time he has access to the respiratory trace. And there is a training phase before doing the actual acquisition. The training phase consists of acquiring B0 field maps in the subjects at different respiratory phase of the cycle, right? Because we know that is going to be different depending on where we are in the trace. So we acquire a couple of these, then we fit a model, and this model will optimize the homogeneity in a particular region of interest, which we can define. And then we know exactly the amount of current that we need to send to each of the amplifier in order to adjust the shim, the shimming in real time with the respiration. So to show you a little bit how it works, this is an example of an EPI. And I'm going to run, so this is a video that basically, it's a short TR, it's about 500 milliseconds, so you can see that it's really moving in real time. The person breathes, and you can see that while the person breathes, the spinal cord slightly moves also in the phase encoding direction. And when we turn on the real-time shimming, we see that because the B0 field is more stable along the respiration cycle, then we have less of this, A to P motion effect. And I told you that I would prove that the motion is not due to a physical motion of the person. We also acquired fast GREs, which are less sensitive to those susceptibility effects. And you can see that while the person is breathing, there is no apparent motion of the body, right? So it's really a B0 variation effect. So this project is ongoing now. We are anticipating the 7 Tesla system coming to Montreal, and we are now designing a shimming coil, like an integrated shim and RF coil for the 7 Tesla system. Okay, so challenge number 3. There's only 4 challenges. So I told you before that there's a lot of QMRI techniques out there. So now imagine that you're going to start a project. Your supervisor asks you, "Oh, can you make a protocol in for measuring myelin and axon diameter in 20 minutes?" Then you're gonna read the hundreds of papers that are out there, and then you're gonna become very overwhelmed by basically what to choose. And if you decide to choose those 2 techniques, what are the acquisition parameters that you need to choose from? So there are already existing consensus for brain protocols with QMRI, but nothing was really established for the spinal cord. So we started this Spine Generic Protocol initiative that involved several centers, and the main goal was really to devise some feasible protocols that use product sequences across the 3 main vendors, GE, Philips, and Siemens. And the protocol is accessible online. It's already accessible through the Open Science Framework website within a standard operating procedure as well. And the protocol is composed of a 3D T1 weighted MPRH for brain and spine assessment, a better resolution scan centered in the spinal cord for cord atrophy measures. Measuring cord atrophy is very important for many diseases, including MS. Having a good contrast between the white and the gray matter. So in the spinal cord, the white and gray are inverted in the sense that the gray matter is inside and the white matter is outside. It needs to be fun, so we need to add some fancy QMR techniques in there. So there is a magnetization transfer. It's in fact an MTSAT protocol, and a single shell diffusion tensor imaging protocol. So all that run between 20 to 30 minutes depending on the vendor. So kind of a pre-validation was to scan someone across different sites. So this someone was me. So I'm lucky enough to travel. And not going to go through all the different sequences, but just to show you what we can get with this kind of standardized approach. This is the result of a T1 weighted, where the spinal cord was segmented using automatic tools that we developed in the lab. And then from this segmentation, we can measure the cross-sectional area. So basically the area of the spinal cord, which again is a good biomarker for many diseases. So when you look at the resulting cross-sectional area across the different vendors, so you have black, blue, and green, respectively, GE, Philips, and Siemens. First of all, you can see that within center, the standard deviation is less than 1, and 1 represents 1 millimeter square. So what you can see is that the standard deviation is actually better, smaller than a single pixel area. So it's quite a remarkable reproducibility. There are slight differences across vendors, and this is due to slightly different contrast, and as you probably guessed, the contrast drives the boundaries that we define for segmentations. So that could explain some of the differences. And in general, the coefficient of variation, the within vendor coefficient of variation was less than 2%. So that's pretty good. For diffusion MRI, we calculated the fractional anisotropy within the white matter at C2 to C5 vertebral levels. And again, the coefficient of variation was less than 2.5%. Fairly good reproducibility within and also across vendors. You can see some red cross from time to time. This is because this project was, going on while still optimizing and adjusting to the specificity of each site in terms of, software version, presence of a particular license or not, and so on and so forth. So it's a difficult translational project, but we're getting there. Challenge number 4. I mentioned about those tools to compute the segmentation of the spinal cord. So that relates to image processing. There have been a lot of developments for brain quantitative MRI data from the UCL group, the Oxford group, NIH, MGH in Boston to compute, to basically process QMRI data of the brain. But so far there have not been a clear package, software package for processing spinal cord data. So in the lab, we have initiated the Spinal Cord Toolbox, which is a comprehensive and open source library of analysis tools for multi-parametric MRI of the spinal cord. And it has a couple of tools, including segmentation of the spinal cord, a template, because you've probably heard about the MRI brain template. There was no equivalent of the MRI spinal cord template until recently, and also processing tools to basically register non-linearly subjects together or to the template. So just maybe one or two features that I'm going to talk about is this template thing. I mentioned that there was no template until recently, so my former student Benjamin Delenaire created this so-called PAM 50. PAM stands for Polytechnique, Aix-Marseille University, and MNI. And the 50 is because 50 subjects were scanned. And he created this T1 and T2 template that is now merged within the same coordinate system as the ICBM 152 MNI template. So it's a nice step forward in terms of being able to have a template, registering new data to the template, and then process and extract particular quantitative MRI metrics within the gray matter, the white matter, or different subtracts of the white matter. Other directions that we are currently working on in the lab. Of course, we dived into this deep learning wave, and the reason we did that is because we realized it was fairly performant for simple segmentation tasks, and especially when we started to have our users, coming to us say, "Oh, we tried your software, and with my patients with strong spinal cord injury, it just doesn't work. The segmentation is leaking everywhere." So, which is why we developed models based on deep learning to basically train segmentation tasks on different pathologies and different MRI contrast, different resolutions, and so on. The end goal is really to make it work for as many centers as possible. So that's an example of spinal cord segmentation. We also tried to segment the gray matter, and this has some implications, some clinical implications when we want to measure, for example, ventral horn atrophy, which is affected in the ALS populations, for example. And so it's really like a magical tool where we could also apply that in ex vivo scans. It was a radiologist who was interested in finding the morphometry of the gray matter in a very high resolution scan, and so he had almost 5,000 slices, which 10 years ago we would have probably done that all manually, and now we can basically do that automatically. So it's quite fantastic. So the spinal cord toolbox is mainly used by people doing functional MRI in the spinal cord, quantitative structural MRI, and also patient population. I would say mostly MS and injuries, traumatic or non-traumatic injuries. So I'm going to give you one example of application, which is a project that was recently finalized. Actually, last week it was accepted in Brain. So it's a project that involved 12 MS centers and 8 neuroradiologists who segmented MS lesions. And I'm going to show you what was the purpose of this project. The purpose of this project was to have all those lesion masks in all those MS patients and register them all together within the similar coordinate system, sum all the lesion masks, and then obtain a probability map of MS lesions in the spinal cord. So we had a rough idea of what was the distribution of MS lesions in the past, thanks to histopathological work, but we did not really have that from in vivo patients. So the goal of this work was really to obtain this probabilistic map and see where do lesions mostly appear in MS patients and how we could explain their disability depending on the phenotypes. For example, people with primary progressive versus secondary progressive, do they have different lesion features and so on. And that could potentially be of interest for people developing drugs because they will be able to target their drugs to very specific phenotype. Okay, so I told you about all those MRI techniques, and I asked you to believe me that we can measure axon diameter and myelin concentration in the central nervous system. But you should not really believe me when I say that, and in fact we should really look at proper validation of those QMRI techniques. So in the last 5 minutes of my talk, I'm going to present you our setup that we've made in Montreal for validation. So basically, get your sample. In that case, it's a cat spinal cord on which we acquire a couple of the collection of QMRI techniques: diffusion, MT, myelin water fraction, and so on. And then the same sample is then prepared for histology, stained, with osmium because we want to use electron microscopy. It's polished. We scan with the EM system. It's a scanning electron microscope system. Then, since we are most of the electron microscopy work when we want to look at microstructure is usually using transmit EM because we can get very high resolution and look at the actual hyperstructure of the axons and the myelin. In our case, it's kind of a difficult job because we cannot play with 2 nanometer resolution and then compare our big voxels from MRI because the acquisition would take years, right? So we need to find a good in between. We used in-plane resolution of about 100 nanometer that enables us to acquire fairly large windows, and then we stitched all those windows together in order to get a full axial view. So it's a couple of millimeters square field of view that we can then point by point compare with the MRI. Then we segment the myelin and the axons on the histology. We resample it to the MRI, we register it, then we can compute, as I mentioned, pixel-wise correlations to see whether, for example, axon diameter measured with MRI corresponds to axon diameter measured with histology. We could also use existing atlases, register those 2 atlases, and then do some track-based statistics. But the main focus I want to look at is the segmentation part, because if we look at what it involves, at the end of the day, what we want is we have this full-scale histology. Here is from a rat spinal cord. So when we zoom in, we start seeing those thousands of axons. And by the way, this is the size of an MRI voxel, approximately. Then if we further zoom in, we really see the axons and the myelin in the transverse plane. And ultimately, our goal is to segment individually the myelin in red and the intracellular portion of the axons in dark blue. And that will enable us to then derive some quantities such as axon diameter distribution, myelin density, myelin thickness, and G ratio. So let's imagine that we're to manually segment all those myelin and axons. What would it take? So let's put that into numbers. The density of axons is about 100,000 axons per millimeter square. The white matter cross-sectional area in this example is about 10 millimeters square. And the time to segment in 2 different labels, axon and myelin, for a well-trained grad student is about 1 minute. So it would basically take 2 years to segment this single image without sleeping, eating, and so on. So that's really not possible. Again, machine learning can come to the rescue. So we developed a tool that is open source. So you can try it if you have some histology work to do. We trained 2 different models, one for SEM, another one for TEM, because the features, the image features were drastically different. I'm just going to show you some results in different organs and resolutions and species. So works fairly well for the macaque corpus callosum with TEM. We also tried it with a human SEM in the spinal cord, a mouse TEM, and also rat SEM. So we are constantly improving this software. So if some of you are interested in working in this field, we are currently hiring. But so to give you some ideas of the benefits of this approach is that when we acquire our full-scale spinal cord, we can then apply this automatic segmentation method. And in each portion of the tissue, we have access to quantities such as myelin thickness and then axon diameter and so on and so forth. Then we can really compare that with MRI. But after we got those results, we thought there could be maybe more exciting things to do than just validating QMRI. And we looked at the textbook literature, and we realized that there was, in fact, a big gap in describing the white matter macrostructure in the human species. So what we did in collaboration with a neuroanatomist in Trois-Rivières was to extract an ex vivo human spinal cord and really do a thorough and comprehensive histology work on that spinal cord. So we extracted it, labeled, fixed. We prepared it for SEM. Then we scanned it. So each segment of the spinal cord was scanned, and then we segmented it with our tools in order to get a comprehensive description of white matter macrostructure in the human spinal cord. We wanted to go a bit further, and we registered it to our MRI template so that now anybody interested in doing spinal cord quantitative MRI techniques have some sort of reference template in a common space with quantities that describe white matter macrostructure. So that's ultimately the goal. Of course, it's only based on one subject, so we could argue that maybe there's a variability that is not taken into account, but that's kind of a first step, and ultimately we would like to move forward with those approaches. So that's kind of summing up my talk. I talked about the concept of in vivo histology with the QMRI. Try to convince you that we could get some useful information, but I also would like to reassess that those measures are noisy. They are not always very accurate. So validation is key, and interpretation is also key as well. Standardization, I think, is very important to ultimately bring it to the clinics and making sure that like anybody in Australia or Quebec would end up with quantities that they could compare. So that's why efforts towards standardization is important, and also open source analysis tools. Basically, try to open the black box of analysis, and if you code some pipelines, try to make the pipeline as transparent as possible so that others can replicate it. In neuroimaging, we have a huge problem of replication these days. So that's one way to address it. And we all have a responsibility towards that. And that comes with communication as well, because computer scientists need to talk to physicists, clinicians, so that they all speak the same language and can understand what is needed and how those techniques work. Clinical translation is difficult, and publishing articles is not enough. When you make a study, you say, well, everything is in my paper, just look at it. It's not enough. So meeting people, training people from different fields is key, and that was our motivation for creating those workshops. If you're interested in spinal cord imaging in general, come to our workshops. We've created those every year, and they come right after ISMRM, so it's one day after ISMRM. And the good news is that in 2019 it will be in Montreal. So if you're around, just pop by, and you're gonna hear some interesting discussions. So with that, I would like to acknowledge people in my lab who contributed to those slides, collaborators, and funding sources, and thank you for your attention.
Thank you. Very interesting talk. Are there questions for the speaker today? Joseph?
I was curious about this dynamic shimmy you mentioned. You said that there was a training phase. So I imagine it's a machine learning algorithm that goes from the pressure down.
Simple lookup table.
Okay.
Right. But we are looking into possible use of machine learning. basically try to because there is some like similar patterns across people. Every people breathe in a different manner. Some more with the chest, some more with the belly. So there are probably things that we can learn from it. But currently we don't try to learn. We just map and then we record the points we need, and then we just map them in our fitting algorithms. Yeah.
Okay. So you do the training at the beginning of every scan then?
Yeah.
Okay.
Exactly.
I have a question with regards to the RF coil that you designed to go with that shimming coil. Now it becomes a sort of a one-sided transmit coil from underneath the spine. So does that, I mean, the spine's pretty close to it.
Yeah.
That's a good thing. But do you find that you're sacrificing image quality when you move to a transmit coil that is one-sided like that versus a bird cage?
Yeah. To the point that we stopped using it.
Okay.
Yeah. So we have now built another coil, which is a receive-only coil, and it's using a more recent technology where RF coils can have some like chokes on the side so that they can pass some DC current without interfering with the receiving part of the RF. So
that becomes a combined
that
shim
becomes
and receive.
a combined shimming, and the nice thing is that the coils are very close to the region of interest, so we can shim.
They're no longer separated by the RF coil.
Yeah, exactly. So we can transmit with the body coil. We can receive with each individual coil array, and we can also shim locally with that. So that's the technology that we are currently working on. So we've built one for 3T, and we are now designing one for 17.
It's a clever design.
Do you anticipate there being any sort of alignment issues with the shim coil? Because I imagine it's just sitting under the patient. If you move it a little bit, everything's kind of off.
So you mean off compared to the calibration phase of seeing what's the profile of the actual shim coil? Because when we receive, I mean, Ryan could tell you that more with the added emotions in the voice. But when we received the shim coil, we had to basically calibrate it and see what would be the generated B0 field per amp, right? So, and that is a kind of that indeed is kind of a static with respect to the physical coordinate system of the MRI. So we indeed had to make sure that this coil was always positioned the same way. Then we had some fear after a year, so maybe maybe it warped, maybe it changed, so maybe we have to redo the calibration. So I guess that was your question, right? Yeah. I know, Ryan, if you want to add on that.
Well, I don't think it's specific to our experiments, but if patients move in an MRI, it kind of messes things up. So, sometimes what happens is we'll do our training phase for the shim, and then there's 5-10 minutes or something, and then by that point, subject becomes sort of uncomfortable and is moving around a little bit. That can kind of spoil the experiment.
What can you do about immobilization of patients in spine? Because that's something I haven't thought about at all personally.
Making them as comfortable as possible.
Yeah, okay.
Yeah.
There's not much restraint used.
I mean, the body is kind of heavy, so it's not like it's like people with head. It's more difficult to to stabilize the head.
Yeah.
For the spinal cord, I would say it's mostly fine. They won't move up and down and things like that.
You don't attach them to the table.
No, we don't. Yeah. Well, what could happen is that they might tilt the head, and that has an impact on the cervical aspect.
Right.
Yeah.
Okay. Thanks again.
Thanks.